Thank you all for your patience. So it's my great pleasure today to introduce Professor Michael Young, a Richard and Jean Fisher Professor and Head of the Laboratory of Genetics at the Rockefeller University, as well as a 2017 Nobel laureate. Prof. Young received his PhD from the University of Texas, Austin in 1975 for his work on Drosophila genetics. He then did postdoctoral research on transposable elements with David Hogness at Stanford University before moving to Rockefeller in 1978. There, he used Drosophila to explore the molecular basis of circadian rhythms. Specifically, he identified clock genes that form a biochemical oscillator within the cells of our bodies, regulating sleep, arousal, and essential daily functions. Most of the Drosophila clock genes discovered by Prof. Young and his colleagues are also central to the circadian pathways of vertebrates, including humans. Recently, his lab has shown that a prevalent human sleep disorder is caused by dysfunction in such a circadian clock gene. Prof. Young's elected memberships include the US National Academy of Sciences, the American Philosophical Society, the President's Council of the New York Academy of Sciences, the American Academy of Microbiology, and the Physiological Society London. Along with colleagues Jeffrey Hall and Michael Rosbach, he received many prestigious awards, including the 2009 Gruber Neuroscience Prize, 2011 Horwitz Prize, 2012 Canada Gairdner International Award, as well as Massry Prize, 2013 Wiley Prize and Shaw Prize, and the 2017 Nobel Prize in Physiology of Medicine for the discoveries of molecular mechanisms that control circadian rhythms. Today, he will be presenting an exciting talk titled Chronic Social Isolation Signals Starvation in the Drosophila Brain and Reduces Sleep. Prof. Young, please.
Thank you for that kind introduction. It's good to be with you. Sorry for the technical delays, but I'm pleased to be able to tell you what we've been learning about social isolation and neural responses to it in this model system that we've used so often in the past, that being Drosophila neurobiology and neurogenetics. So I'll start by telling you a little bit about some human data. Part of the reason we became interested in this problem and wanted to focus on Drosophila is because of work that had been published not very long ago by University College London, in which there was a study, a year-long study starting in March of 2020, just as the pandemic was beginning. And it's a study of about 70,000 UK respondents, and the respondents were asked to rate their sleep over the course of a year periodically. On a 5-point scale, very good sleep was given a high rating, and very poor sleep a low rating, a 1 versus a 5, for example. And what you notice in this graph going from March of 2020 to March of 2021 is a slow erosion of the perception of the quality of sleep by this large cohort of individuals in the United Kingdom. And it was true for both females and males. And of course, you can see the timing of many of the lockdowns in the UK over that period of time. So it's quite clear that sleep has eroded, the quality of sleep has eroded over that year-long period. And of course, the stresses and strains are many, which are related to responses to COVID. But one of the major components of that stress is social isolation. Now, we can't really measure how much of this response is due to a response to social isolation having a direct effect on sleep in human individuals, but we can ask the same kind of question in a model organism. And again, that would be Drosophila. So we've tried to ask in Drosophila whether Drosophila respond to social isolation. And if so, if one of the responses to social isolation involves their quality of sleep. Now in Drosophila, as you see over here on the right, we often use a fairly simple monitoring system, which we refer to as a Drosophila activity monitor on the right side of your screen. And the idea is that flies are loaded into individual tubes, one fly per tube, enough fly food, solid fly food at one end of the tube to keep these flies alive for several days or even a couple of weeks. And these tubes are placed in monitors, of which we have hundreds. Within those monitors, there is an infrared beam produced by an LED that is followed with a series of phototransistors that indicate when beam breaks occur, which are indicators of the movement of the flies within these tubes. All of that digital data is collected by a computer. And over here on the left side of the screen, we see that this is turned into a readout in which we can see the individual behaviors of many, many flies. Here we have a stack of about 20 flies, each being recorded over a 24-hour period. Daytime is indicated as 0 to 12 in the light open bar. The dark bar indicates nighttime when the lights are off. And what you see is each fly is showing a pattern of behavior that is largely shared by all the flies in the group displayed here. So flies are fairly active in the morning. They have a noon or a midday nap, which is extensive. They become active again toward the end of the day, and then they show consolidated rest, consolidated sleep at night. And this can be transformed into another representation here, showing sleep amounts varying across the day and night. So this is the way we can measure sleep and activity in the flies, and then ask questions about behavioral stresses. So again, our question is, does social isolation reduce sleep in Drosophila? And the way we approached that was to take a bottle of flies in which we have dozens of flies together for 3 to 5 days to give them the acquisition of social experience. And then we remove the flies from those bottles and put them either 1 fly per tube, 2 flies per tube, 5 flies per vial, 25 or 100 flies per vial. And we hold them in that situation for a total of 7 days, and then record their sleep-wake behavior using the activity monitor as I described previously. What we find is there is an erosion of sleep in the individuals that are in isolation, that is where we have a single fly in a single vial. We find that significantly diminishes sleep, primarily daytime sleep, but surprisingly, even 2 flies in a vial eliminates this issue. 2 flies, 5 flies, 25, and 100 flies are indistinguishable with regard to the level of sleep that they maintain. It's just the single fly that's showing this diminished sleep response to social isolation. A second question we wanted to ask is whether social isolation is perceived over time. Is it isolation duration dependent? To do that experiment, again, we gave the flies an opportunity for several days of social experience, and then moved them to vials in which we either had 25 flies or a single fly in isolation. And then we maintained flies in that configuration for either 1 day, 3 days, 5 days, 7 days, and looked after each of those durations at their sleep-wake behavior using the Drosophila activity monitor. What we found was that there was a progressive loss of sleep, primarily daytime sleep, that arose as the flies were isolated for longer and longer periods of time relative to the group-reared flies. Importantly, we saw this response not just in a limited number of strains of flies, but we saw it in many Drosophila species, every species we tested, many isogenic strains, young versus old flies, and in several sleep inbred panel lines. These are panels of flies that were recovered from the field. They're wild caught flies, but in establishing inbred lines, we find that they can have longer or shorter duration total sleep over 24 hours. And in every case, whether they were long or short sleepers, they behaved in the way shown here, social isolation having a profound effect on the amount of sleep when it becomes chronic, that is 7 or more days. This just breaks down the information a little more extensively. For example, total sleep continuously erodes over 7 days. Daytime sleep, as I pointed out, erodes more severely over those 7 days. And especially sleep occurring in the first few hours, the first 4 hours after dawn, that's where we see the greatest amount of sleep. So this is the early portion of the midday nap that the flies are having. This seems to be the most robustly affected by social isolation. Now, we've asked whether there are tricks that we can perform to try to relieve this social isolation. For example, if we take a single fly and put it into a vial where previously many flies have been reared, presumably there would be odors left over, chemical cues left over in that vial from the prior rearing of a number of flies in that vial. But this has no effect on the level of sleep loss compared to a single fly in isolation for 7 days. We've also inserted a glass slide in the middle of one of these vials where we have a number of flies on one side of the slide, a single fly on the other. So the lonely fly, we'll call it, can see the other flies but can't touch or smell them. That fly still acts with respect to sleep as if it's lonely. If we have a perforated separator so that the animals are able to smell the live flies on the other side and see them, that also, if anything, exacerbates the sleep problem. Even a little more sleep seems to be lost under those conditions. And if we use a still more permeable filter between the group and the lone fly, we also get, if anything, a diminution of the amount of sleep versus a lone fly, indicating that it's not just being isolated, but perhaps the feeling of not being able to join a group. This sleep loss is relieved by allowing another species of fly. If we add numbers of a small number of flies of a different species, Drosophila of a different species, this will relieve the sleep problem from the lone Drosophila melanogaster. And we get a variable response from adding other kinds of pets, if you will. 2 different strains of ladybugs. One strain seems to have no effect, but the other strain seems to improve things. So this needs to be investigated more closely. Other kinds of insects can have either no effect or a further diminution in the responses to isolation. So we have a number of questions that we have that arise from these kinds of experiments about what is being perceived here by that lone isolated fly. Now we're most interested in the neural responses. What in the brain is responsible for these responses? This is something that we certainly can't ask about. We don't have the tools to ask about this in humans, but in flies, we have many tools to more deeply investigate the neural basis for these stress responses associated with social isolation. We reasoned that since most of the sleep lost is in the early part of the midday nap, that if we looked using RNA-seq for RNA changes, gene expression changes that were occurring immediately preceding that part of the midday nap. In group reared flies, chronically isolated flies, or in acutely isolated flies, that meaning flies that were isolated for just a single day, that perhaps we could compare the gene expression patterns found in those three groups and determine if there are gene expression changes that are unique to these chronically isolated flies. So we compared the chronically isolated flies to group-reared flies, and the isolated 7-day isolated flies, chronically isolated flies, to single day of isolation, and look for something that was unique to the chronically isolated flies. We found 274 genes that fit that pattern. Now, when we ask what's the nature of the genes that are affected in these gene expression assays, we're interested in two kinds of changes. We're interested in changes that occur in the chronically isolated flies, the 7-day isolated flies, that differ from both the group and the 1-day isolates, where they look about the same, but we have a strong diminution in expression in the chronically isolated flies, or alternatively, where we have a strong induction of new gene expression uniquely in the chronically isolated flies. And when we put these two groups together and do GO analysis, gene ontology analysis, to find out what kinds of genes are being regulated or now misregulated by social isolation, we find that most of the responses, the most significant responses we get by GO class, involve metabolism, involve genes that are involved in the metabolism of the fly. Now, if we look further and say what are the most robustly changing gene patterns in the family, we have a list of top 20 changes, most of which are upregulations in the genes shown here. There's one exception, a downregulation, which is for a gene called Drosophila. This is a gene that produces a hormone that responds to satiety. When the animal is no longer hungry, this hormone goes up. And what you can see here is the hormone is reduced following social isolation. Up here, we have a gene called Lymostatin, which we know is an appetite stimulating hormone. This is upregulated in these socially isolated flies. On the right, you see a series of arrows, and those arrows point to genes that were previously identified in a study of gene responses to 24 hours of starvation. So what we see here is a pattern emerging from social isolation that resembles that of a starving Drosophila brain. Quite a surprise to us, since we began by studying sleep. So given that gene expression pattern, we wanted to know if behaviorally, in addition to affecting sleep, social isolation was affecting feeding in these flies. Now, to do this, we required a new device, something called an activity recording capillary feeder, or ARC for short. And in this setup, we have a fly in a tube. 3 tubes are shown here with flies in them. And in each tube is a capillary in which liquid food is placed. And what we now have is video monitoring of both the activity of the flies and the position of the meniscus on that liquid feeding tube. So as the meniscus moves, we can measure feeding, and as the fly moves, we're measuring sleep versus wakefulness. And on the right, what we see is what happens in the sleep profile and in the feeding profile. And you'll notice that once again, sleep is lost in the chronically isolated flies. And here what we find is feeding is enhanced, is accelerated, is upregulated in the chronically socially isolated animals. And this is more of a quantitative analysis of the same experiments for total sleep, for daytime sleep, nighttime sleep, and again, ZT 0 to 4 hour sleep, that early morning sleep, what we see is essentially the same response, the same loss of sleep at each of these episodes that we showed with the other style activity monitor. But now if we look at feeding, we see total food consumption, daily food consumption, nighttime food consumption, and early morning food consumption. And all of these are significantly upregulated. That is, the amount of feeding that we see, the amount of food consumed at all of these times is about twice that we would see in group-reared flies. So there's a profound effect on the amount of food intake by flies that are socially isolated. So they're losing sleep and they're eating more. Now, this is reminiscent of a recently published article by the American Psychological Association. This covered a year of the pandemic, essentially the same year that I spoke about in the University College London study. And the summary of this study is that about 60% of American adults have either lost or gained weight that was unwanted over the period of the pandemic. Now, weight gains are shown on the right in red, weight losses are shown on the left. And what's clear is that most individuals whose weight is changing, these are weight gains rather than weight losses. It's about 2/3 to 3/4 of the population of weight changes are in the gain category. And you see from the types of individuals that have been looked at that this is pretty uniform across all segments of the population, and the size of the weight gains is really startling. 30, 40 pounds gained in a single year, for example, in many of these individuals. That's an average weight gain in many of these individuals. So in many ways, our model organism Drosophila is doing very much what many of us have been doing over the course of the pandemic when chronically isolated. Now, I want to go back for a couple of minutes and cover, once again, look at some of the genes that were strongly misregulated by social isolation. And I want to focus on Lymostatin, which again is a starvation-induced hormone, as I mentioned before. This is a hormone that previously we did not know was expressed in the brain. It had previously been shown to be produced in the corpora cardiaca, which is outside the head. It's part of the thorax. And so we decided that we would use immunoside chemistry to see if we could locate the cells that must be giving this Lymostatin signal in the brain of our socially isolated flies. And what we found is there is a Lymostatin signal in the central brain. This is antibody to Lymostatin, and it involves a structure called the fan-shaped body, which is in the central brain, the central complex of the brain, and is has been previously associated with a number of behavioral functions ranging from orientation, learning and memory, and sleep regulation. So Lymostatin is produced in these cells. The cell bodies fall outside the fan-shaped bodies, but their projections go to the fan-shaped body, to a layer in the fan-shaped body. And importantly, what is shown here are signals from GFP that are driven by a reporter, by a driver and reporter system, which we call P2. These are P2 neurons that are lighting up here. Their projections are lighting up here. P2 neurons had previously been associated with a center of the brain that was responsible for regulating sleep. So importantly, if we merge these images, we see that there's a strong overlap in the projections of both of these neurons, that the P2 neurons and the Lymostat expressing neurons are closely mingled in their projections. And moreover, the connectome that has been produced at Genalia, or Hughes Genalia Research Center, have recently shown that there are extensive synaptic connections between these two groups of projections in the fan-shaped body. So we know from the connectome analysis that these cells are capable of interacting with one another synaptically. We've asked what happens if we suppress the activity of the P2 neurons. Can we block, for example, the response to social isolation of reduced sleep? What's shown over here on the right is a driver driving in P2 neurons a potassium channel called KIR2, which when expressed will cause a neuron to become inactive, no longer electrically active. When that is expressed in P2 neurons, what we see is that the 7 days of isolation no longer has an impact on the sleep of the fly. The controls of the two parental strains that were used to make these flies, which don't themselves express the necessary combination to inactivate the P2 neurons. So inactivation of P2 neurons will suppress this response to social isolation. Will also repress the feeding response to social isolation. So here are the controls showing the overeating of chronically isolated Drosophila, and down here the suppression, the same suppression of those P2 neurons in these individuals brings these closely aligned with one another. So we've essentially removed the effect of social isolation on appetite. We were also interested in what might happen if we activate the same neurons. Is it sufficient to activate these neurons to get a stress response in the fly that is comparable to that produced by chronic social isolation? And I just want you to focus on this bottom corner here. What we've done is we've expressed a temperature responsive cation channel called TRPA1. When exposed to 28 degrees, a warm temperature, that channel opens, and that will cause neurons with that channel to become active. At 22 degrees, the channel is inactive. What you can see is at 22 degrees, group reared flies and flies isolated for a single day both show the same response, which is in a lack of the activation of that channel. In contrast, when we activate the channel either in group or isolated flies, only the flies showing a single day of isolation show a response that indicates that they now are feeling chronically isolated. There's a diminution in sleep in response to activating those neurons, but only if they've seen a day of social isolation. The group-reared flies don't do this. We get the same response with feeding at 22 degrees. Group-reared and isolated, one-day isolated flies are the same. However, when we raise the temperature to activate those channels, one-day group and one-day isolation, those now differ, with a day of isolation producing the overeating phenotype that we saw previously. So activating these neurons can produce a phenotype resembling chronic social isolation, but only if the flies have at least experienced a day of social isolation. So it somehow makes them miscalculate or misperceive the duration of their social isolation. Now, once again, I just want to point out that there's a convergence of neuronal projections from Lymostatin producing neurons and sleep promoting neurons. R23E10 is a driver that activates neurons in the central brain that are known to be sleep promoting neurons. And here we're looking at anti-Lymostatin, 23E10 or GFP. And now when we merge them, we see that they are in close contact with one another. And as I said previously, these neurons from the connectome analyses that have been performed are extensively synapsed to one another. So we think there's a direct interaction between the feeding and sleep neurons in this discrete region of the brain. So let me just finish by drawing some conclusions. Drosophila can distinguish between acute and chronic social isolation. Chronic social isolation activates central brain neurons that regulate appetite. Increased feeding, or the starvation brain state, induced by chronic isolation appears to broadly reset gene expression programs that are associated with metabolism. Again, that was a surprise when we first saw that. The drive to feed may inhibit sleep. Of course, if you must eat, you can't sleep, and if you must sleep, you can't eat. You could imagine that those two things could have an impact of one another. Nevertheless, the axonal projections of previously documented sleep-promoting neurons are extensively synapsed with projections of the P2 neurons that are involved in this response to social isolation. So we feel there can be a much more hardwired regulation of this balance between sleeping, gain of appetite, and loss of sleep. Finally, hyperactivity of P2 neurons alters the duration of social isolation needed to reduce sleep. And this suggests to us that the P2 neurons somehow encode a time-dependent isolation status. So I'll just finish by pointing out the people that did this work. This is a party we had a few years ago in Sweden. This is Wan-He Lee, who is the principal scientist on this work in the lab. And she was a postdoc in the lab. She's recently become an assistant professor at Texas A&M University. And Zikun Wang, formerly a graduate student in the laboratory, also very much responsible for the work done on this project. Thanks for your attention, and I'm happy to take questions.
Thank you for the very engaging talk. I don't see any questions at the moment. Let me know if I'm mistaken, but I can start with one to get the ball rolling. So basically, it seems that isolation might induce this kind of a state of stress in the animal. That could be one hypothesis. And are there other studies from your lab or other labs which look at the effect of, for example, acute versus chronic stressors, perhaps shock or some other kind of stressor on sleep and appetite, and whether that's mediated by similar or different mechanisms?
Right. That's an important issue that we need to address in the future. It's related to a question that we're addressing now, which is, does social isolation, are these neurons that we've discovered, is there activity necessary for the perception of the stress of social isolation? Are they no longer stressed? And that's why we no longer, when we inactivate those neurons, is that why we're no longer losing sleep and no longer overeating? Or are we simply blunting, are the animals still feeling stressed, still perceiving their isolation, still stressed, but unable to respond appropriately or in this fashion to that stress? And so one of the things that we're doing in that case is to look at other responses to social isolation. So for example, one of the responses that we're aware of is aggression. So social isolation increases the aggression of flies. And so we're interested in understanding whether those aggressive responses, the responses of aggression, are also linked to the P2 neuron set, for example. Again, the idea being that if everything, and there are other responses that we can look at as well. And the question, the question we have in mind is, if a whole series of different responses to social isolation are all eliminated by suppressing P2 activity, then this begins to sound to us much more like the animal's not perceiving the stress anymore, or not perceiving it in the same way. So this is sort of the first step in that kind of a direction. You've asked about whether there could be other stressors that show up chronically, and we haven't gone down that road yet. We thought social isolation is a form of stress that's easily provided without producing on the face of it important physical damage to the fly, like for example concussion or some kind of a shock treatment, as you'd mentioned. But we could certainly go in that direction in the future.
Right, thanks a lot. I see more questions now. So one is from Lockyi Louis Choi. Some has suggested the contribution of metabolic problems to the development of cancer. Sorry, you have to scroll back again. Given social isolation will alter metabolism, will this in turn contribute to cancer development? Or could this?
Yeah, and of course we don't know yet, since these studies are fairly early on. We're just getting the first look at what's going on. But certainly if we look at human populations and the response to unwanted weight gain, we know that diabetes, for example, is associated with unwanted weight gain, and that there are interactions between diabetes and predictions of the incidence of certain forms of cancer. But these are all important questions to ask, but we don't have any more along those lines in the fly. But again, we can point to the expectation that if the fly is in fact a good model for what goes on in humans following social isolation, that there could be a variety of metabolic responses that are contrary to good health.
Great. And there's another related question by Leo Chan, very interesting as well. Are there changes to microbiome composition and activities due to isolation-induced sleep disruptions, or maybe due to isolation in general, which then might exert these neuronal changes through this gut-brain axis?
Yeah, we're asking that question now. The same question has arisen in the principal scientists that have been doing this work. And we don't know the answer yet, but I think it will be interesting on two levels. Our first pass is just to ask, in the whole fly, are there any changes in commensal organisms that we see in response to social isolation, whether they're in the digestive system or elsewhere? It's quite possible that there's a change in the load of microbiota all over the animal. So it'll be very interesting to see if there are changes and where those changes are. And we certainly haven't excluded the possibility that there could be changes outside the digestive system that are important, in spite of the fact that appetite changes do suggest a careful look at what's going on in the digestive system. So hopefully in a year's time, we'll know much more about that.
Right, I can probably take a few more questions. Which ones? Okay, there's a top voted question by Sumina. Are the effects of chronic social isolation on the brain and metabolism reversible upon regrouping the flies? Basically, can you rescue these effects? Yeah. Yes,
it seems like you can suppress these rather quickly by moving a lonely fly back into a group. So they are reversible, although one of the concerns about a pandemic, for example, is that we're talking about 1 or 2 years of impact versus 7 days of impact. And of course, you could say, well, 7 days out of the life of a fly might be comparable to 1 or 2 years in a human life. But it does suggest that for flies and for humans, that these should be reversible changes.
Yes, and another very interesting question. So these flies were previously social flies. They were previously exposed to social cues. So Tingwei is asking, what happens if they were isolated from birth, I guess,
Yeah,
all the way
yeah.
through? Then are there similar or different effects? Yeah.
Yeah, we don't know that yet. The way we would like to do that is by collecting fertilized eggs and raising them throughout their early development by themselves, so that they never see another fly, and then asking what whether isolation of that sort produces a different effect. And of course, the control for that would be to have a group of embryos that are put together in a comparable vial, let them grow up at the same in the same time frame, and see if there are differences in behavior of the sort that we've seen in the experiment so far. But it's an interesting problem that we're anxious to follow up on.
Yep. Well, the last question is, well, the one on the chat is similar to what has been asked before, so I don't think I'm going to ask that, but I guess, is there any more? Um, okay, there is a pointed question. To what extent can these findings be extended to humans? And
We don't know, but it certainly suggests that Drosophila has been a great model for many areas of human biology. For example, we have a long history of working on circadian rhythms and the genes and proteins involved. And those genes and proteins that were originally found in Drosophila allowed us to bootstrap into mice and humans. And it's essentially the same mechanism in us for circadian rhythmicity as it is for Drosophila. So we use Drosophila to give us ideas about how and where to look, what kinds of changes may be occurring, and then it makes it much easier down the road. I think you wouldn't immediately jump to humans, but you might ask whether or not similar centers of regulation, similar changes in appetite and sleep occur in a mammalian model. Mice, for example, which are so readily genetically worked with. But certainly, as I showed you, we've got large numbers of observational studies, large numbers of individuals in observational studies, human observational studies, that indicate weight gains and lost sleep over the course of the pandemic. And I suspect that social isolation is an important player in those responses.
I agree. I studied the zebrafish, and we also see effects of isolation on various kinds of behaviors. So, and to get my 2 cents worth is, I think anything important in biology is probably going to be very well conserved across many different species. So I think we can wrap this up right now. So thank you so much, Prof. Young, for this very engaging talk. And yep, hope to see you in person one day. And thank you all for coming.
Okay, thank you. Bye-bye.